Thank you. Namaskara.
Yes, okay, namaskara.
Good afternoon. We have with us our esteemed KSTA Chairman, Honorable KSTA Chairman, Professor S. Ayappan. On behalf of all of us joined here, I welcome Professor Ayappan sir to this webinar. We have with us session chair, Professor H.S. Savitri Madam, who was the head of biochemistry department in the Indian Institute of Science, and she is the esteemed member of KSTA. On behalf of all, I welcome you, madam. We have with us esteemed member of KSTA, former Vice Chancellor of Gulbarga University and BLD University, Professor Basaraj Moolimani sir. On behalf of all of us, hearty welcome to you, sir. And today's esteemed speaker, Kirthana Sindhe, is with us. Dr. Kirthana is the Commercial Marketing Manager and Customer Experience Lab Research Solutions, Merck Life Science Private Limited, Bangalore. She will be delivering a talk on drug development. On behalf of all of us, hearty welcome to you, Dr. Kirthana. I also welcome all the participants, especially few students and few lecturers who have joined this webinar, both on WebEx platform and also on YouTube platform. Many will join on YouTube platform because some students say YouTube is quite easier to join. That's why many students join on YouTube. For all of them, hearty welcome on behalf of KSTA. Now I request session chair, esteemed member of KSTA, Professor H.S. Savitri Madam, to take over and conduct the session. Over to you, madam.
Thank you, Dr. Ramesh. It's my pleasure to welcome all the panel members and our Honorable President Ayappan and all the participants of this webinar for this very important and exciting talk on drug discovery and drug development. Drug discovery and drug development is truly an interdisciplinary area of research involving bioinformatics, biochemistry, chemistry, and then cell biology and structural biology and computational aspects of biology and pharmacology. You name it, almost it embraces all branches of science. So in that sense, drug discovery and drug development is a translational research, and we are indeed very, very happy that we have a very eminent researcher in this area, Dr. Kirthana Sindhe. She is a cancer immunologist and has had 11 years of experience in drug discovery and drug development. We couldn't have asked for a better speaker for this important topic. Dr. Kirthana obtained her BE degree from in biotechnology from Mysore University, and then she moved on to get her MTech in pharmaceutical chemistry. You see, from biotechnology to pharmaceutical chemistry in DIT, and then she obtained her PhD in the area of molecular biology and genetics from JNCASR, Bangalore. So you see, right from BE to PhD, she already had equipped herself in so many different disciplines which are required for drug development. Molecular biology and genetics is another aspect I forgot to add into the disciplines that are involved in drug discovery. For her PhD, she worked in the area of malarial pathogenesis from the year 2009 to 2015. And then she moved to United States for her postdoctoral research in the University of California, San Diego from 2015 to 2018. Here again, she moved from molecular aspects of pathogenesis to discovery or methods of discovery of anti-malarial drugs and target identification. So indeed, she has taken a logical route to drug development and drug discovery in the course of her training. Then, with all these expertise, she worked in the area of drug discovery and development of therapeutic antibodies for cancer and COVID-19 as scientific manager in the company called Zumtor Biologics from 2018 to 2021. Then currently from 2021, she has been a commercial marketing manager in the Customer Experience Lab, which is a leading where she is leading and also overseeing the Merck Center, CSIR, and a high-end skill development in the Merck Innovation Center at Bangalore. So she has had extremely wide experience in this area of drug discovery and development. I am looking forward to her talk, and I'm sure you will enjoy the talk as much as I will. Thank you. And now I request Dr. Kirthana to deliver her talk.
Most of. Am I audible?
No, audible, please.
Yes, madam.
Okay. Thank you, ma'am, for actually saying everything. You articulated my entire career journey very well. And you, in fact, understood how I've shifted the fields and how I've tried to be diverse, which covers various topics, yet focused on drug discovery and drug development. Thanks to Dr. Ramesh and Ayyappan for giving me this opportunity for presenting today to the students and other faculty members. So I will start with my talk on this topic. So before we get into the details of the main topic, I just want to give you a little introduction about our Merck labs. So here, right now, I have joined Merck, and if you see here, Merck is a vibrant science and technology company. We are present in 66 countries. We are leader in life science. We're a vibrant team of specialists in healthcare, life science, and performance materials. We're dedicated to solve the toughest problems in life science by collaborating with the global scientific community. In this line, so Merck established two labs, which is called Merck Innovation Lab at Bangalore and Merck High-End Skill Development Center in collaboration with CSR MTech in Chandigarh. So the vision has been to support, enable, and facilitate life science research in India by providing tools and techniques. So the talk today would focus on the skills as drug discovery and drug development, which we enable through our labs as well. And these are the different working areas of the lab which actually falls into the drug discovery development pipeline. Like as ma'am mentioned, it is interdisciplinary subject. So all of these working areas connects to the entire process of drug discovery and drug development, which we currently focus in our lab. So to begin with, let's understand what's drug discovery. It's a process by which new pharmaceutical candidates are discovered. So a pharmaceutical candidate, it could be a chemical compound or it could be a biological compound. So it's mostly having a pharmaceutical activity. So that's how when you discover a new compound, we call it as drug discovery because you discovered it. Drug development, as the name suggests, you're developing that molecule. So it is a process of bringing a new pharmaceutical drug to the market. Once a lead compound, you identify a lead compound first, and then you bring that into the market through this process of drug discovery. So it's important to know why we should study this topic. So to understand why it is important, it is a very complicated and expensive process. So it becomes important even before we do a discovery and development, we understand the entire process. What are the challenges present and how expensive a process is, which gives you a clear-cut idea whether how much should you invest on a particular compound and also prioritizing the compound which should go into the development pipeline. So here, as I mentioned here, it takes 10 to 15 years for a company to bring out the new medicine into the market. So you may ask why it takes so long. I will be covering in the next coming slides. But as you know, it's an important process for the clinical researchers and also the company itself to see how you could bring the right candidate to the market. It's not just a process that you discover and you just bring it to the market, but what is its intent? Is it really a safe product? Is it really useful for those target patients? Are the patients getting benefited? So that's why it's really important to understand the entire process of drug discovery and development. As today we also have the major audience as students here, so I will also touch upon how it is very important to your career as well. So not just a concept in science, it's also important how you could choose different careers in this particular field. So there are different stages in drug discovery and drug development. First itself, as name suggests, drug discovery, you're going to discover a molecule. Then you enter into a preclinical stage. Clinical trials are there. We have different clinical trial phases that happens in human. We have next FDA approval that has the shortlisted candidate will be approved. And then there is also something called post-market surveillance. That is, you're still monitoring the drug, even though it is approved and is it in the market, you still see, because some of the human population might respond very differently. So this is why the post-market drug safety monitoring is very important. And you might have heard a lot of big blockbuster drugs get withdrawn even after they were approved because it started showing a lot of side effects in certain population. So if we go now a little deeper into drug discovery, there are many ways you can do drug discovery. So first, you could identify a new molecule, but they could also be you identify a variant of an existing molecule. So you might have already a very well-known molecule, probably you want to tweak a bit in that molecule, and certain properties you can improve, and that can become another very good drug for intended use. So identifying a molecule that acts on a specific biochemical pathway. So as we all know that human beings, we have a lot of biochemical pathways that is guarded within our cellular processes. So most of the molecule, it will hit the different biochemical pathways, and you could design a molecule to that kind of specific biochemical pathway. Or a natural compound which is extracted from plants can be, or different organisms can be obtained. Or repurposing, there's already an approved drug, but you want to repurpose it for a different indication. For example, if you've identified a drug, let's say for Parkinson's disease, maybe you now that drug you could repurpose it for another disease, maybe Alzheimer's disease. It's just a hypothetical example I'm sharing. There's also been a lot of repurposing when you identify a molecule for, let's say, melanoma cancer. Such molecules also get approved after certain years to a different type of cancers, maybe a lung cancer. So that is what is called repurposing. So it's very important during the drug discovery, you identify a disease. Which is the disease you're going after? That's when you start looking out for a potential targets, which you could identify through genomics or proteomics approach, that where you will understand, okay, this is the drug and it is actually targeted to a particular protein or a particular gene. So that's how you identify a potential targets. And then you start screening the compounds. You might have already synthesized certain compounds based on the disease and the targets which you have identified. There are different ways of actually bringing out those new compounds. It could be through chemical synthesis, natural products you extract through natural sources. There's also target-based design. That is, you already know a target, so you're going to fit the molecule onto the target. So as ma'am mentioned before, bioinformatics. So that gives you it's one of the good to chemoinformatics, and bioinformatics gives you a great tool to understand how those but through in silico approaches, you can actually see how these molecules are interacting with the drug with the targets. So you could visually do a lot of docking studies to understand how your molecule fits into the target. And these days, it also gives opportunity to screen multiple compounds for even the solubility, stability, absorption, metabolism, transport, if whether it's reacting with other drugs or other off targets. So all of that information now can be easily available through chemoinformatics and bioinformatics. So the research has really changed from what it was 10 years back. And then once you have shortlisted a group of compounds, you start screening them, virtually screening or high throughput screenings where a lot of robotics are there. It's automated screening. And then you bring out the shortlisted compounds. As I already mentioned, there's molecular modeling, which you could do, and you can start qualifying the compounds which you have shortlisted. So that is where we call a lead compound, and the lead compound now enters into different studies. So now if we come into drug development part, you will see preclinical testing, which generally happens either in vitro or in vivo. So in vitro is you conduct in the labs in cell lines. So in vivo is in the animal models. So these are the closest to the human. Why we do in animal models? Because your next phase of study is in humans. So before you put your new drug into human, it's very important to see how it works on animals. So even within the animals, there are different models. It ranges from rats, mouse, it also goes up to monkeys. So a lot of these animal models are employed to understand different processes of drugs. So why do we use these animal models? So before it enters human, you want to understand whether that new compound, what you've discovered, it is safe, it is efficacious, that is, it is really working for the cause that you've created for, it's not toxic, and also to understand the ADME process. What I mean by ADME is absorption, that is how well your drug gets absorbed, and distribution. So you want the drug to reach to the target site. So how does it get distributed between the tissues and reaches to the site of target? And metabolism, how much time it takes for the drug to stay within the body, whether it gets metabolized faster, because if it gets metabolized, it has to stay for a certain duration of time for it to give a response in the body. So it's important to know what is the metabolism profile. And then finally, the excretion, that is elimination. It should not even stay for too long, otherwise there will be toxic effects as well. So the ADME profile of a drug is very important, and it is being evaluated in animal models so that you make certain calculations here, and then you, as per the clinical protocols, you then administer into humans. So in phase 1 study, where we now start evaluating safety. The first phase 1, it's mainly for safety, and we see a small sample size. It could be 20 to 100. And we also try to evaluate whether you should administer drug once or a repeated administration, whether it is effective, whether it's being tolerated. All of these parameters are checked in phase 1 trials. In phase 2 study, now we're increasing the population. So now you've found that in small size, all of these parameters are good to go. And now we go into phase 2 study, where you do in 2 groups, you have control groups. You also have the target groups, the patients who you want to treat, for which the drug is being designed for particular disease indication. So there are 2 groups, and now the sample size is increased. We're going to test in 100 to 500 people, and it could be a double-blinded study. That is, neither patients nor the researcher who's conducting the study would know with whom which product or is being injected into these two patient groups. So that is to evaluate whether the person is who receives this compound is responding to that drug. So as you heard of placebo effect, even if there was no drug given, still the patients respond. Some of the control groups responds. So that is why you have a double-blinded study to understand how this molecule works in two different population, and also you start measuring the actual safety, whether it is effective or not. Now, once the drug reaches phase 2, we now go into phase 3. Now you're increasing the population even more. It's now large study. It's very well designed. It needs to be very thoroughly designed. A protocol has to be thoroughly done because you cannot repeat this study again. You have so many thousands of patients who have enrolled. The enrollment itself takes a long process, shortlisting through inclusion, exclusion criteria. So I'm not touching those details because that is another whole session to talk about. But it's important to know that the phase 3 clinical trial is the last step in clinical trials. It's done on multiple patients. Sometimes multi-center study is conducted, again double-blinded study. And once you have evaluated its efficacy and safety in this larger population, it is then the drug is filed for the FDA approval. So in FDA review process, you have safety and effectiveness being shortlisted there. You review all the data. So there will be a guidelines given by FDA that your drug has to pass through all of those criteria. So you will be submitting all of those documents, and there's also inspection of the clinical study site when you're conducting the clinical trials. So there's a lot of follow-up, very in a very scrutinized way. The data will be evaluated, and that's when you get an approval for the drug, and it then enters into the market. So in post-marketing drug safety monitoring, there will be a review, monthly or yearly reviews. They'll keep seeing the dosage and usage information. There will be reports which would be generated very periodically to understand whether the drug will stay. So any drug that makes into this biggest process that's why it's called a successful drug. It went through all of these challenges, and it's staying in market for a very long time. So this is the entire process of drug discovery and drug approval. And you will see that how many compounds actually start with in drug discovery stage. There are thousands of compounds, and as you move through the different stages of drug discovery, drug development, there's only one compound that gets approved in that big pool. And you will see the money invested in throughout this process is more than 300 million per compound, and it takes roughly up to 15 years to bring out one drug. So you will see a lot of pharmaceutical companies, they have a big drug development pipeline. They have all the lead compounds shortlisted, and as and when it goes through this process, they will start seeing which one should go forward, which one should stay. So now coming to, there could be compounds which fail, right? So I said just one compound comes out, and imagine if that compound fails. So it's really important that we take care of it very, very much at the beginning, because you're spending so much time and money to bring it out. And what could be the reasons for the compound to fail? These are the 4 top reasons why the compound fail, and it's related to the drug's ADME process, which I said: absorption, distribution, metabolism, and excretion of that compound in the body of the human being. So it's important to see what's the pharmacokinetics properties of the compound, whether there's animal toxicity, adverse effects, and if you have developed a molecule, maybe it is safe, maybe it's no more toxic, and it has a very good pharmacokinetic properties. It's very well tolerated in human. But what if it is not at all effective? it's not giving that much benefit to the patients, then again the compound would fail. So that's how these are the different reasons why the compounds fail, and it's very important that we get thorough with this process and understand the right kind of methodologies to employ to avoid failure of a compound and to avoid the big huge expense in terms of time and money. So as I mentioned, to a lot of here in the audience, we have students who look forward for what kind of opportunities would be available for their career. So if you look into the job market and opportunities, this drug development pipeline is where very important for the life science field. As I already mentioned, these are the different phases, and your zone of opportunity is mostly here. So when you start with your life science, your graduation it could be any field in life science. It could be biology, chemistry. It could be within biology, molecular biology, cell biology, bioinformatics. So you will see that most of you would be getting your skills, and you would be falling into this zone where you would get into drug discovery and development up to the preclinical phases. So it's very important that you identify, as per your field, what is the skill set that is needed and how you should align your skill sets to these different pockets of drug discovery and drug development. And then if there are a lot of population who get interested in the other side of the drug development pipeline. So once you have the core scientific background and expertise, you can start layering upon your skills for the phase 1, phase 2, and phase 3 studies, and even post-market surveillance. So that's how you will see that your current job opportunities it spans in the drug discovery and drug development pipeline. So it's important to understand each step in that, start accumulating skills and knowledge through your subjects, and start seeing where is that you want to align yourself in this process. And then when you are ready with your skills and knowledge and your degree, you will then see your job opportunities in these phases. So you could start looking up what kind of roles are present for you once you are entering into this market. So that's what I wanted to give a brief background. So I'm giving a little an example of Keytruda, which is a biologics. So now I mentioned that you either would have chemical compounds or you might have biological compounds. So what I mean by a biological compound is antibody. So antibody therapeutics has become very attractive. The reason being it is very specific for the particular disease. So you must have heard in treatment of cancer. Earlier, there was a lot of chemotherapy which was in practice. The field started with chemotherapy treatment. And what we were facing is a lot of side effects. The patient would go through, and there was no option other than chemotherapy. The patient had to go through chemotherapy. But in past many years, there is a shift in the field. Now we use biologics, and especially a field which has evolved is immunotherapy within the targeted therapy. So in immunotherapy, what happens is you're activating your body's immune cells to fight cancer or fight any disease. So that is how this examples comes from, which is called Pembrolizumab. I will be explaining how actually it works and how it was being discovered. So this was approved in 2014. This was the first approval, and now you will understand how it is being done. So the generic name of it is Pembrolizumab, and it is a programmed death receptor 1 blocking antibody. So it is to treat certain cancers like advanced melanoma, non-small cell lung cancer, head and neck squamous cell cancer. If you see that here in this cartoon, you have your immune cell. So T cell is part of your immune cell. We have different immune cells in the body, T cells and B cells and different subsets. And we also have NK cells. So here an example, if you see the T cells, which is shown here, and then you have a cancer cell, which is in purple. So most cancer cells, they express these targets, which is shown here, PDL1, PDL2, and then your T cells, it has PD1. So what happens is when these two interact, and the cancer cells, when it interacts with your immune cell, it will block the immune cells to bring out its action. So what happens is the cancer happily grows because it's not letting the immune cell to kill it. So you need to block this interaction. So this interaction is now blocked by this antibody, that is pembrolizumab. It will come and get block this interaction. So now the immune cells can kill your cancer cells. So this is a breakthrough in cancer treatment, that is anti-PD-L1, PD-1 therapy. And you will see that it's now pembrolizumab was FDA approved in 2014 for advanced melanoma, whereas in 2015 it was approved for other indication of cancer, that is in lung cancer. And as the years passed by, it was approved for various other indications within the cancer. So this is how you can repurpose the same drug for different disease indications. This is an example of Keytruda, and you will see its development timeline, though here I mentioned till 2017, but you see there's multiple approvals for Keytruda. And I also mentioned about chemotherapy. So now Keytruda is also being used in combination with chemotherapy to bring out even more best results in patients. So you will see that the recent approvals for Keytruda is in combination with different other antibodies, different other chemical compounds, and it is bringing out a lot of benefits to the patient. And one important example to show here is the triple negative breast cancer. So there are three types of breast cancer, which is like hormone sensitive breast cancers. You have triple negative breast cancers, and they are biggest challenge. The triple negative breast cancers are very challenging to treat, and Keytruda found a place there, and it was used in combination with chemotherapy, where now you can tackle the early stage triple negative breast cancer. So that is how the field has progressed, and Keytruda has got multiple approvals in its development timeline. So another important resource which I want to share with all of you is if you want to get, if you're interested in understanding certain trials, so clinical trials, as I mentioned, your drug actually starts from preclinical studies and enters into the clinical trials. You can actually start, you can go to the clinicaltrials.gov. All the current clinical trials are listed there. So you can type in any compound or dates that you want to understand, and you will get the data retracted for those particular compound or a particular trial name if you already know. So this is how the trials would be, you would be seeing here. You have different, all the details of the trials is mentioned here. You have study details, eligibility criteria, conditions, you have study identifications. So this will actually help you understand the entire how the trial is conducted, what are the results of the trial, what is the status, the recruitment status. As I mentioned, patients are recruited for certain trials. So that is how the entire trials are conducted, and you can start looking into these. So this is one example of Keytruda which I mentioned. I can now go back to drug discovery and drug development process. If there are any questions in this part of yes, in this part, do let me know if there are any questions in this part before I proceed. And we will also have a Q&A at the end to understand more. So here now, as I move forward with this part. As I mentioned, our skill, we at Merck Labs, we are providing the skills that actually fall into drug discovery and drug development process. And we have our center LinkedIn group where we provide a lot of training programs. Currently we have students, faculties, all of them who have joined our LinkedIn group, and we have 7,000 plus members in LinkedIn group, and we provide different skill development trainings here. And yes, so I would end our presentation here because I want to spend more time in giving addressing Q&As. So you will see that we provide trainings, hands-on training programs in our labs, and we follow COVID safe protocols, and we have a lot of students passing through these trainings, and this is their happy faces where they got trained and they're willing to go ahead and make a difference in their career. So I will stop the presentation so that we could make this more interactive, and I can take all the questions that you may have for drug discovery and drug development.
Thank you very much, Dr. Kirthana. Now,
You're welcome.
Thank you, Kirthana. Yeah.
You're welcome, ma'am.
Yeah. So Ramesh will find out whether there are questions that are to be addressed.
Yes.
Ramesh.
Are there any questions, Mr. Anthony? Madam, actually, these are PU students. They are a bit shy to ask questions. Sometimes
Okay.
they will mail the question. If they make, we will be forwarding it to Dr. Kirthana.
Okay. Okay.
And
Yes, I have, sir, as you mentioned, you wanted this to be more towards the students who want to understand basic concepts. So I have made this in line with that. So they could enter in the chat box for a Q&A if they would like to have more understanding, or you could also send me the questions.
I had a question regarding this skill development program.
Yes.
How do they apply, and what does it cost? Like, is there any fees or some such thing? And what is the duration and where it is conducted?
Yes, sure, ma'am. So as I already mentioned, we have two different labs. One is Merck Innovation Lab in Bangalore, which is in Electronic City, the Bommasandra Jigni Link Road. And we have another lab, which is in Chandigarh, that is in collaboration with CSIR MTech. So what we do in these labs are hands-on trainings. So let me also share a brochure so that you would, and also to all the audience who are here. So I can share our lab brochure so you will get more clear picture on what we actually do. Able to share the content. Okay. Unfortunately, I'm not able to share the content.
And what stage are these skill development training given? Should it be somebody who has had a BTech, or is it at what level these trainings are conducted?
Sure. Are you able to see my screen?
No, not yet.
Okay.
Umesh and Anthony, please help, madam.
Huh?
Umesh.
There are no questions, sir.
No. Dr. Kirthana wants to share her
screen.
Okay. No, please check, ma'am.
Now I can. Yes, now I can. Yes. So to address the question, so this is our labs, as I mentioned. So we have different lab working areas. So we have molecular biology, functional genomics, mammalian cell culture, protein biomarker detection and screening, and proteomics. So these are the working areas and what we offer in skill development programs. So we have digital trainings and workshops. So you will see these are the different topics that we provide trainings in like digitally, and we also have hands-on workshops. So now you as there are two bifurcations. There's basic training programs, there's also advanced training programs. So basic training programs are for the students who are new to the field, who want to start acquiring those first skills in a particular field. So that's how we have these basic programs. Once you have developed those initial skills, you can sharpen it and through our advanced training programs. And we have also listed all our advanced training programs. So it is open to students right from they start graduating at bachelor's, and it is up to even the PhD scholars who want to acquire certain skills for their research work. It's mainly, as I mentioned, our labs, we want to actually provide the skill developments for their for having boost their employability and also to drive the innovation. So it's two purpose: developing your scientific competency and advancing your techno and bringing out the advanced technology. So the pricing of this program, so the good news is, as I already mentioned, join our LinkedIn group. So there are a lot of free webinars that we give every alternate Mondays. So I will also paste that link into our chat box so that you can join our LinkedIn group, where we give a lot of free webinars. So you can take advantage of those. And we also provide these digital trainings, the certificate course. So if the students are interested in providing certain certificate course, they can also join through, we have different pricing for different training programs. So anyone interested can get in touch with us and we can provide a particular pricing for these programs. And again, coming to the hands-on training programs, as ma'am was checking for the pricing, it starts from, for the basic programs, it starts right from 7,000 to, it goes up to max, 10, 15, 20,000 based on which training program you use. But the quality of our programs are so good. We cleared your concept, we build that scientific competency, we give you a full hands-on training with all our instruments in the lab. So you will see here the instruments that we have for biomarker. We have high-end instruments in our lab, right from Luminix MacPix, SMC Pro, where you can detect the biomarkers at the femto level. So these are the different instruments for biomarker discovery studies. We also have Snap ID for Western blotting, the high-end equipment where you can do Western blot within 30 minutes as compared to the traditional approach, which takes 1.5 days. We do a lot of gene editing techniques. Students who are interested in functional genomics. We have mammalian cell culture. We do automated cell counting, imaging of the cells, all of these 3D. We are currently establishing 3D cell culture facility. And we have recombinant DNA technology, helping in cloning, production of recombinant proteins, purification of proteins. So, so I hope I could address ma'am's
Duration.
Sorry?
Duration of the course.
Duration, yes.
Yeah.
Okay, so duration, it starts from 3 to 5 days, and it can go as long as maybe 15 to 20 days. For some of the students for digital training programs, what happens is they have colleges and they say we cannot attend the whole day. So we have been very flexible. So we ask them 2 hours in a day, are you able to spend? So we customize it and we give them the digital training programs every day 2 hours. Sometimes we extend for over the span of 10 days or 21 days. So that's how we do. And hands-on training, minimum we do 5 days so that students get a very good hands-on with their pipetting. They understand how they should use the pipettes, how they should use different instruments in the lab, and how they should conduct experiments, how they should troubleshoot experiments. So it starts from 5 days. For experiment in advanced trainings, such as mammalian cell culture assays, or let's say, take recombinant DNA, these takes a little longer. So it will go up to 10 days to 15 days, where students can come and learn these techniques. So we are very flexible to customize it. So our basic program starts for 5 days. We do not want to be in hurry. We want students to get those, and we let the students touch, feel, and experience our instruments. So that's the one of the differentiator that we have. We do not restrict our students. They are welcome to use all our instruments when they sign up for these training programs. Yeah, I
Dr.
hope.
Kirthana,
Yes.
I have just a little small inquiry. How
Yes.
the medical universities can collaborate with Merck and send their MBBS and postgraduate students some kind of training there.
Yes. Yes, so medical and pharma students also we have been training. So it depends on what they are looking for in career. If they are looking for a career where these working areas are coming in play for their career, like I mentioned, molecular biology, cell biology, if that is coming in play, or if they are having a certain projects, let's say clinical studies, they're doing a lot of clinical studies, and most of the time it's the biomarker detection that they do in clinical studies. So if whether they want to get trained into clinical studies where how to detect those biomarkers, or even gene testing profile. For example, during COVID time, we had real time qPCR because RT-PCR became mandatory for most of us, and that technique, I think now even a layman now understand what is RT-PCR. So that's how you can, based on what research project they are interested in and where they want to make their career shift. So based on that, we can customize the training programs, and they can choose among these working areas what is that they want to do. So we are also in future encouraging certain thesis dissertation projects for long term, so that students can benefit. And we also offer internships. So every
Okay.
quarterly, there are 2 internships that we offer, and we give, it's a paid internship. So we
Okay.
pay students. It starts from 15,000 to 20,000. So student can apply for our internship. So every 3 months once they can start applying, and they come and they get trained in our labs.
This collaboration with universities in the sense of recruitment of patients for trials in particular region.
Okay, so that is more towards patient side. So I'll have to check with our internal team whether we could participate in those, but we can definitely provide technical services or technical support for those kind of trials. If you have identified that this is your study and this is the outcome you're looking for, then we can help you with inputs, intellectual, scientific inputs, and also technical support. This is the technology that you could apply for this study, and we could collaborate in those terms and help you out in that.
Thank you.
You're welcome, sir. We have been signing a lot of MOUs with colleges across India. So as I mentioned, we want to build life science skill in India. So that is why I think in next 5-6 years, we want to spread throughout India, and we want to build that skilled workforce so that the students are confident to make their career choices. Because right now there are colleges and certain universities who are limited with infrastructure, and our students cannot get that kind of benefit, and they're not in par with those universities which has these infrastructure. So that is how Merck's vision is. We give that equal opportunity so that students can enable their skills, plus they can make a better career choices. So far, we have trained 12,000 plus students and research scholars, covering almost 1,000 universities and colleges so far. Our dream is very big. Our goal is very big. So we really want to reach out. And I think through KSTA, it's a great opportunity that I got connected to all of you and also students, so that they can get benefited from this program that we offer, and they can get best use out of this.
So KSTA also has quite a good network with all the colleges. So maybe they can pass this your talk to various colleges so that they get
Sure.
to know that such a such a innovative center exists. Yes.
Yes,
And we
truly,
can
ma'am.
make use
Truly,
of it.
ma'am.
Yes.
Truly, ma'am. And also, ma'am, in our training, apart from core skills, like core technical skills in science, it is very important we also groom students for facing the opportunities, how to even find the opportunities in science, how they should prepare for those opportunities. So a lot of career guidance program is also integrated into our different skill development programs, so that the students are bold enough to move forward and make the decision. This is what is my vision. I want to become a scientist in so and so subject next 5 years, and this is the right skill that I want to acquire, and this is the way I'm going to acquire the skill, and I am ready. It's not that once they finish their degree and college, I have heard the pain points of students. They go into a lot of depression, that where am I going to get that job, whether they are underconfident. So those are the pain points that we want to address through our programs, through the interaction with Merck, giving them the new hope that they can get going in their career.
Our Honorable
Yes.
Chairman,
Professor Ayappan had a
Yes.
question.
Thank you,
Yes.
madam. Thanks for this opportunity. Through you, I would like to immensely thank Dr. Kirthana Sindhe. Ever since we have met her, we know what kind of an inspiration she is and the kind of motivation she brings to youngsters, particularly for taking to science. And then looking at the lab that the Merck has in near Bangalore, we are proposing the MOU, which is getting almost in taking shape. And in the next coming EC meeting, we would be placing it as a supplementary agenda item. I'm sure you all would approve that MOU so that we can move together faster for the benefit of the youngsters in and around Bangalore. Thank you, madam, for this opportunity. Thanks,
Thank
Chairman.
you very much, sir. Yes, I'm looking forward for the collaboration.
Thank you, madam.
two of our esteemed KSTA members, Professor S.K. Saidapur and Professor Ambekar Eknath sir are there. Sir, hello, Saidapur sir, Eknath sir.
ಇಲ್ಲ ಏನಿಲ್ಲ ನಮಗೇನು ಪ್ರಶ್ನೆ ಇಲ್ಲ.
Hello.
Hello, please sir. Hello.
One of your slides really got me. Can you hear me?
Yes, sir. Yes, sir. I can hear you.
Really got me wondering.
Mhm.
It showed a figure of $300 million to produce one drug.
Yes.
Now, a poor country like India
Mhm.
is able to sell drugs. We call it generic drugs or whatever, but at the cheapest drugs in the whole universe is in India.
Mhm.
For example, I used to carry drugs for my friends in Indonesia or in Philippines or in other places too. Can you
Mhm.
tell us what is the secret behind India's approach to drugs and the cost of drugs?
Okay. Yes, I think you rightly mentioned that developing country like India, it's really hard to pump in such an amount of money. So what happens is there are different lead compounds in the drug development pipeline. I think the clinical trials is what actually comes to a big handy. That is, you evaluate in such a way that you understand which is the molecule that's moving forward. So there could be sponsorships. There's government sponsorships for clinical trials. So that way you could bring down the cost of the study. And the pharma company, which you see mostly in India, they're not more into novel development. There are few startups, there are few companies who are investing into novel compounds, but it's mostly the existing drug gets repurposed. And also you have big pharmas who you invest huge amount of money. So what the secret is that as you move into clinical phase trial and you start showing this molecule is working wonders in this particular indications or patients, you attract different companies who will invest into it. So that is how some of the startups work. But the big pharma who have already been producing a lot of drugs, they have the drugs stay for a longer time in the market, and they would have really made those sales and got back their profit. So they pump in the money into the next drug, which is in the development pipeline. So in that way, it's a cycle. You invest, you return, you get returns, you invest, you returns. And then when you invent a new drug and it becomes very attractive for other investors, that's how your cost would be reduced and you have different people sponsoring your study. So this is based on my understanding. I could answer. These are some of the secrets. I hope that was.
Also, many of the drugs that are already in the market, their patent, would have expired. So in India, they can make it cheaper and sell
True.
it. That's how the cost of some of the drugs are low in
Yes.
India, because it is because the patent rights are
Yes, correct.
expired. So they can now synthesize and sell it at a cheaper rate. Yeah.
Yes,
Thank you, ma'am.
true.
Dr. Kirthana,
Yes.
these are PU students. After their exam, after CET, some of them would like to enter into biotechnology. They would be interested in having some orientation courses, days orientation, like in astronomy, the Jawaharlal Nehru Planetarium. They will be conducting orientation program for PU students.
Okay.
Like that, is it possible to have an orientation course online in association with KST?
Sure, sir. When we talk about our collaborations, when we are laying out our MOUs, we could have certain programs that way design, and we could see how our faculties availability also. So based on that, we will definitely try to do such arrangements as well.
A second,
That's a very good suggestion, Ramesh, mainly because many students who are in PUC will want to know what is there in store if they take biotechnology, B.Tech, or
Yes.
for example, BSc biotechnology, whatever. So it will help them to understand or orient whether it's really a field that they should go for and things like that. Yeah.
Yes. So as I mentioned, we give a lot of career guidance programs to students, right, through our training programs. I did not flash that slide today because it was more on drug discovery and drug development. So I did not bring in that piece. However, I told students that their zone of opportunities lies within the drug discovery, drug development pipeline. It is in the discovery phase and early development stage. All their skill sets are needed there, and that's where they have opportunities for job. So I think I can also quickly pull out just for your curiosity that you carry today. I think I'm also curious to share that to give you that how I did not bring it today, but as Dr. Ramesh was saying, during the Science Week, they have a program where I thought I could share that. So I'm quickly sharing that just for the curiosity sake. So here, okay, so you can see my slide. So this is how I have depicted here. You see the entire field in biotechnology. So it starts from R&D, the discovery, and it goes all the way till regulatory affairs. And there are other non-R&D areas like commercial operations, other operations and services. You will see that different fields that branch out with science as a skill, and the people who with scientific skill and layering that skill with a different other skills are making them very attractive in those jobs, and they're getting very specific jobs. So and definitely there's a lot of tons of opportunities for biotechnology students or students in life science. I would say I did not bring that today in my presentation. I thought I would reserve it for the future Science Week program, wherein I can elaborate the entire opportunity that students have in life science. I think when I graduated, I was also at the fear where I was not clear. Plus, I was in the fear that what opportunities I have. But I think I'm happy to share that based on my own experience through, because I had a breadth of experience from bachelor's, plus how the field has changed so overall. So I would be able to give that overview, the 360 degree view, what kind of opportunities are in store for students.
Dr. Kirthana, one more request. You said on alternative Mondays you'll be organizing talks. You can
Yes,
share with us, KSTA can
yes,
share for
sure.
also social media.
Sure, sir. I will share that with us. So right now we are doing mammalian cell culture talks. So there is one talk coming this Monday. So I will share the link with you.
We will be sharing to all the universities across.
Yes. So I
Yes.
think I will also, if all students are there, I will also try to paste that link so they can actually join our LinkedIn group, where we also conduct a lot of activities, science activities, quizzes, and also we keep sharing a lot of events. So this is our LinkedIn group. So all of them who are here, they could join us, where you'll get updates of all our upcoming trainings and programs. And the webinars, the free webinars, which I was talking about, it happens alternate Mondays right now. They can join through that group. And I will also share with students how LinkedIn is the biggest platform for their career. It's the biggest professional network, and they should start building their personal branding right from they are at the college level. And over the time, they can groom themselves so well. They are very attractive rather than you going and asking, can you give me a job, or can you give me this opportunity? When you build yourself so much, you become so attractive that opportunity comes and it will knock your door. So that's how I encourage students to build that personality that way, that you are being called for rather than you going and asking for. So those are some of those tips that we share through our training programs. And yeah, I look forward to share all of that in future.
Thank you.
You're welcome.
Madam, move to session chair, Professor Savitri, madam.
Yeah, it was a very nice, wonderful talk, and I'm sure we'll have continued interaction with you and with
Yes.
the Merck Innovation Labs. Maybe I would think that KSTA can organize the talks and the skill development programs addressed specifically to those who are in their final year of
Yes.
BSc or final year of MSc, so that that's where it is set in. PUC is a
Yes.
bit too young,
Early, yes.
too early for them.
For
So
POC, it's more about learning the
career
knowledge plus
development, yeah,
and career,
knowledge.
and giving them the direction,
Understand.
which way they need to go ahead.
Yeah, sure. So which branch they want to go for their undergrad program, such kind of opportunity, career development talks and suggestions would be useful.
Yes.
But serious training should encompass people who are in their final year of BSc or BTech, as well
Yes.
as MSc. Yeah.
Yes.
Yeah. Thank you very
You're
much for this
welcome.
wonderful talk, and we look
Welcome.
forward to further collaboration. Yes.
Thank you, ma'am.
Now I request our esteemed members of KSTA, Professor Mulimani sir, SK Saidapur sir, Baker Kirthana sir, all of them are here for their words. Mulimani sir. So,
No,
ಇನ್ನೇನಾದ್ರು ಹೇಳ್ತೀರಾ sir?
no, it was a very, both scientific aspects of drug discovery and all that. At the same time, the educational aspects from the career point of view for the students, whether in BSc, whether in medical sciences and other things, a beautiful thing. And definitely, one more talk, which can be, kind of
Enabler for students.
arrange it, yeah. And and Dr. Kirthana, I mean, any organization or institution should contact your Bengaluru office or.
Yes, they can get in touch with me.
That's
I'm heading those labs, so they can get in
Okay.
touch with me.
Yeah,
Yes.
that's good. Thank you for a real great talk as well as an interaction. Thank you very much.
Thank you. You're welcome, sir. Thank you very much.
Eknath sir. Hello, Eknath sir. Okay, Dr. Kirthana, it was really a very learning experience even for me, a biologist and an aquatic biologist. This genetics and drug development we had studied in our bachelor's and master's. I had forgotten. It was a recap of the whole thing. Your talk was really very scholarly and insightful. I'm sure students will be highly benefited by your talk, and some of them, I'm sure, will be interested to join biotechnology field as a career for their future. Thank you very much. We
Yes.
look forward to
Thank
signing
you.
you very soon.
Thank you so much, sir, for inviting me for this talk. It was a very warm interaction with all of you. Thanks, students, for listening to this talk. I really hope it has benefited you. You can reach out to us if you face any difficulty through Dr. Ramesh. You can express your challenges or anything that you want to enable yourself. He will help you out through us, how we could help you in this journey of your science. All right. Thank you so much, sir. I will stay in touch with you.
Thankful
Thank
to
you.
session chair, Professor Savitri Madam. Only yesterday I requested Madam to chair the session. She agreed immediately. We are
Thanks.
grateful to you, Madam, for your support and encouragement. Forward to
Yes.
your guidance. And I'm very thankful to Professor V.G. Mulimani, sir. He has been there with us in every talk. And for this talk,
Thanks.
also he has joined. Thank you very much, sir, for your gracious presence. I'm also thankful to Professor Saidapur, sir, and Dr. Ambekar Eknath, sir, for their presence in the talk. We look forward to guidance from our esteemed members for more interaction with private companies as well as government entities in future. Thank you very much. I also thank all the participants who joined on Cisco WebEx and YouTube platform. We look forward to continued engagement with us, JSTA, for your career development. Thank you. Thank you very much.
Thank you.
Thank you. Thank you very much.